Hello my students, how are you all? Welcome back to my EduSpace. Hope you all are doing and learning well. If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. Let's talk about today's video. It's all about the historical background of immunology, how the field of immunology has evolved. So without further ado, let's get started. Going deep into the history of immunology, we have to know about what is an immunology. It is basically the study of immune system. So immunology is the study of immune system and it is very important branch in the field of medical as well as biological sciences. The word immunology has been taken from the Latin word immunis, which means examined. So we can say that the immunology means you are protected or exempted from any kind of infection. The immune system protects us from infectious disease which are caused by pathogens through various lines of defense. We know that our body has different kinds of defense systems. So our skin, all the mucous membrane and the chemical that are present are forming the first line of defense. When this first line of defense is breaches, then our cellular type of immunity has started to work. So when the first line of defense has breaches, then the second line of defense are on call to start or producing antibodies. If the immune system stop functioning, then what happened? It will lead to different types of enormous types of diseases that may be autoimmune diseases, allergic or infections, and can cause cancer also. Immunology. The field of immunology has grown on one hypothesis. There was an idea about the field of immunology to grow, and actually it was observed that those individuals who once got infected and recovered from that infection were thereafter protected from the same infection. Suppose if this is a person which once get infected with eye pathogen. Then further, if the same eye pathogen try to infect the same individual, then this particular individual is found to be protected. Why this is so? Because in that particular person, he got immunized. He got immunized from its prior infection. So this is the basic idea through which the field of immunology has evolved. So we can say the field of immunology has grew on one hypothesis. And what was that hypothesis? It was observed that a person who once infected from any kind of infection, so it was protected from the same infection, or we can say that a person got immunized or immunity to that particular infection. So it is the picture of Thucydides. Thucydides was a historian of Peloponnesian War, which gave the first reference about immunity. He wrote about plague infection in Athens. It killed millions of people of Athens. He wrote that those who recovered from plague infection could nurse sick persons because they wouldn't catch the infection again. So again, he used that hypothesis. We just learned about it that those persons who once got infected with a particular type of pathogen got immunized or got prevented or exempted from that particular pathogen if it catches that particular pathogen again. So after this discovery of immunity, scientists got an idea how we could induce immunity against any pathogen. So this is the picture of Athens, the Peloponnesian War, where millions of people were killed due to the infection of plague. And that time, this basic hypothesis has played a very important role in nursing the people who was actually got infection of plague. After the reference of Thucydides' work, there is another turn take place in the field of immunology, that is variolation. So in this, Chinese and Turks was first performed induced immunity. They performed induced immunity against very fatal disease, which is smallpox. The symptoms includes very high fever, headache, and rashes all over the body that leave scars or misfigured the whole body. This smallpox was caused by variola virus. So here they used dried crust of smallpox pustules and inhaled through nostrils or inserted in cuts of the skin, and this technique was successful in preventing the smallpox. So you can see in this picture, this is the symptom of a typical smallpox disease where the body has got lots of pustules. So these pustules containing pus. It is basically a small blister or pimple type on the skin which containing pus. And what is pus? The pus arises due to infection. It is basically a thick liquid produced by body which consists of dead white blood cells, bacteria and tissue debris and also serum. So they used this type of pustules to get induced immunity. So they used dried crust of smallpox pustules and they inhaled through nostrils or even got inserted in cuts of skin. This technique is now known as variolation and it became successful in preventing smallpox. Work of Edward Jenner. This is the picture of Edward Jenner. He had beautifully used the variolation technique. In other cases, the Edward Jenner in 1798 observed that milkmaids who had once infected with cowpox were immune to very deadly and fatal disease, smallpox infection. So what he observed that the milkmaids who had once infected with cowpox, which is rather mild disease as compared to the smallpox, were immune to smallpox. After that, Jenner inoculated cowpox pustules into people, which ultimately protected them from severe smallpox disease. Instead, they might got infection, but that was very mild and they recovered and became immunized. So this type of technique is known as inoculation. So what he had done, basically, first he observed that the milkmaids who used, the milkmaids, he thought that, he observed that, suppose this is milkmaid who got contracted with the cowpox disease. Okay, this particular milkmaid has got infected once with the cowpox disease, but later on, if this particular person is infected with smallpox infection, then he beautifully recovered the smallpox infection and recovered. Okay, so why this happened? Because this particular person is got immunized through the cowpox infection. Thereafter he got protected from the smallpox disease also. So this was the Edward Jenner's inoculation technique, where fluid from a cowpox pustules were inoculated intentionally to people to get immunized against fatal smallpox disease. So after smallpox, the world had got new disease of cholera, where Louis Pasteur had succeeded in developing immunity against cholera. So this is the picture of Louis Pasteur. What he had done, basically, let me tell you, he once cultured the cholera bacterium. So he once cultured the cholera bacterium. So these are cholera bacterium. This is cholera bacterium. Okay. He once cultured the cholera bacterium, but after returning from summer vacation, he injected chickens with that old bacterium culture. So once he returned from his long summer vacation, what he had used the same cultured bacterium, that is cholera bacterium, to infect the chickens. So now this culture become old, and he injected chickens to that old bacteria. Okay, he found that the chickens became ill, but they recovered. He found that they become ill but recovered. Okay, these chickens were got ill, but they recovered from that cholera bacterium infection. Now after that, Pasteur grew fresh bacterium culture, but due to limited supply of chickens, he injected fresh culture to the previously injected chickens. So he thought that why shouldn't I grow a fresh culture of cholera bacterium? So he again grow the fresh culture of cholera bacterium. But the supply of chicken was very limited, so he again injected the fresh bacterium to its old chickens. But surprisingly, he observed that chickens were completely protected from the disease. So after the infection of the fresh culture of cholera bacterium, all the chickens that were previously got injected with the old bacterium culture were all alive. So he confirmed that aging had weakened the virulence of pathogens. What is virulence? Basically, virulence meaning the ability of pathogen to cause disease. So he confirmed that the aging could weaken the virulence of pathogens, and that attenuated strain might be used to protect against disease in the form of vaccine. Latin vacca. Vacca means cow. So in honor of Jenner's work on cowpox inoculation, Louis Pasteur named that type of immunized form as vaccine. So here is the development of vaccine by Edward Jenner. With the help of Edward Jenner work, and this type of vaccine was produced by or invented by Louis Pasteur. Pasteur again extended his above said findings. He vaccinated a group of sheep with heat attenuated anthrax bacteria, that is Bacillus anthracis. So what he had done, he had first formed the heat attenuated. I'm using the term attenuation again and again. Do you know what this mean? Basically, attenuation means weaken the pathogenicity or virulence of pathogen. So here he heat attenuated the strain of Bacillus anthracis. So this is culture of heat attenuated Bacillus anthracis that cause anthrax. Now he injected this type of heat attenuated version of Bacillus anthracis to a group of sheep. These are the group of sheep. These sheep were artificially induced with heat attenuated anthrax bacillus. So it means that they are induced with artificial immunity. They are induced with artificial immunity. So he then again vaccinated virulent culture of Bacillus anthracis to new group of sheep as well as to the old heat attenuated vaccinated group of sheep. Now what he had done next, he then used the virulent culture of Bacillus anthracis. This is virulent, 100% virulent strain of Bacillus anthracis. Okay. Now he used this virulent culture of Bacillus anthracis and induced to that previous old sheep group and also to the new group of sheep. Okay, this is new group of sheep which were not ever get the infection of Bacillus anthracis. So now these are induced with the virulent culture of Bacillus anthracis. Now he observed that the old group of sheep that was once infected with heat attenuated strain were all live, and those who were never ever got the infection of Bacillus anthracis or heat attenuated version or a new group of sheep, they all died. They all died. So he confirmed that attenuated form of pathogen could be used to get immunized against disease. He confirmed his experiment further that attenuated form of pathogen could be used to get immunized against disease. So this type of sheep was live and this type of group of sheep were dead because this particular sheep was once got the infection of heat attenuated Bacillus anthracis that evolved the immunity towards the virulent version of Bacillus anthracis. So in 1985, he first vaccinated a boy, that is Joseph Meester, with attenuated rabies virus, and he lived as per his expectations. So this is the first experiment which was conducted on a human, and that boy name was Joseph Meester. That's all were the history behind the evolution of immunology. From here onward, emergence of the study of immunology and discovery of vaccines took place. Thanks for watching.